Well, special thanks to the organizers for an excellent symposium. It really reflects the wonderful diversity of the university and its ability to contribute to this really incredibly severe pandemic. So very quickly, when a virus infects a cell and commandeers the replicative machinery of that cell, the viral proteins are processed, modified, ubiquitinated, and then finally go through the proteasome and are loaded co-synthetically on class 1 HLA molecules that traffic to the surface and there serve as an immunosurveillance target for hopefully a capable CD8 T cell that through its TCR recognizes the virus peptide in the context of the HLA molecule and kills the infected cell. Now, the molecules that are encoded by each HLA or MHC allele differ in their amino acid sequence, and the critical part are these pockets that are formed by the amino acid sequence of the HLA allotype that confer different binding properties on peptides. So in this case, this peptide is tethered at this position at the second amino acid and at the ninth, the terminal C-terminal amino acid. And each of these different HLA allotypes look at a different repertoire of peptides. For example, HLA-0201 looks at peptides that have leucine at position 2 and valine, A3 leucine and lysine, HLA-B27 arginine at position 2 and lysine. And each allotypic MHC molecule then preferentially binds a different set of peptides, conferring an entirely different complexion to that individual's immune system response against, in this case, the coronavirus. So our working hypothesis is that individuals with HLA allotypes capable of binding only a small number of peptides encoded by SARS-CoV-2 genome consequently have a limited repertoire of T cells capable of recognizing these peptides. They'll make an impoverished, exhausted immune response that results in that patient's progression to severe disease. So there's precedent for this, and the first precedent comes back several decades ago to when the HIV virus was rampant and there were no effective antiretroviral drugs. So here's an example of how different HLA types see the HIV envelope protein. So for HLA-B27, you look at the envelope protein in terms of peptides that have an arginine at position 2, then some 6 other amino acids, and then anything of lysine, arginine, tyrosine, or leucine. And I admit this is certainly an oversimplification, but it's the essential feature. The B27 allotype can bind then 15 different peptides in the HIV envelope. Look at B35. The B35 allotype looks at peptides in terms of those that have proline at position 2 and tyrosine at position 9. There are none such peptides in the HIV envelope. B7, an intermediate number. And of course, the intriguing thing here is that people who are HLA-B27 are elite responders or long-term non-progressives, so that the average time in the absence of antiretroviral therapy for them to progress to AIDS is about 14 years. In the case of B35, individuals who can't recognize on that particular HLA allotype envelope proteins, the rate of progression is about a year. It's not clear that that is due to this specific envelope recognition, but it certainly serves to illustrate the consequences of peptide recognition. Now, in the case of SARS-CoV-2, If one looks, and this is a study just published by Grafoni et al., this is my quick summary of a portion of that. If we look at different HLA allotypes, A01, A02, a very common one, A3, 1102, 2301, and 2402. And these 6 allotypes are of special interest to individuals who are developing vaccines and things like that, because they, by and large, cover the vast proportion of the human race. So if you want an HLA-restricted vaccine, one looks at these allotypes. From our perspective, if we look at these allotypes in terms of what they can recognize, the proteins that they can recognize from the spike protein or the entire genome, in the case of HLA-01, 4 peptides in the spike protein can be recognized, 54 in the genome, odd different numbers, sometimes as high as 78 peptides for HLA-02, but A2402 is of interest in that it sees only 3 peptides in the S protein and only 14 encoded in the total genome of SARS-CoV-2. And this is an example, just quickly, of what the spike protein looks like, and each of the colored areas show the region that is identified by different HLA allotypes as being that peptide being able to bind. So it covers not only quantitative, but also different portions of the spike protein and indeed the entire genome. So there's a qualitative aspect of this also. Of interest, if we look at the geographic variation in frequency of individuals with the HLA-2402 allotype, it's intriguing that it varies considerably across the human race. In China, Han Chinese, about 31% of individuals will have A2402. In Northern Italy, 24%. Germany, a bit lower, 17, 19. USA varies in different parts of the country. Spain, 17%. England and Ireland, much lower. And of course, we're familiar with the death rates in areas like Italy that are greater. Could it be simply a correlation with a susceptibility allele, either A2402 or other alleles yet to be identified? It remains to be seen, but it certainly is an intriguing possibility. So the aim of this project is to determine the association of COVID-19 disease severity with HLA genotypes. So the SARS-CoV-2 peptide binding characteristics of allotypes associated with susceptibility will be determined in an effort to identify the regions of the proteins that are recognized by those making clinically effective or ineffective immune responses. An additional motivation is to provide necessary HLA restriction information to proposed studies that we're collaborating with Adam Moore and other individuals on the identification of immune cell subsets and their potential developmental trajectories in advanced COVID-19 infection, to really look at T cell exhaustion, cytokine storm development, and other key features of the disease. So we need HLA to understand and to develop the appropriate reagents in the case of tetramers. So what we initially proposed to do is to obtain intermediate resolution HLA typing for A, B, C, and DRB1 loci. We would do this with PCR amplification followed by sequence
minute mark.
oligo hybridization to arrays of probes using the PCR SSOP method. It's relatively fast. It's certainly a limited alternative to high resolution exome sequencing, which is ideal, but cannot be done in such real time. So there'll be 4 study groups, and with approximately 100 in each group to deal with the power and significance of the data. So that numbers between 42 and 82 for some various risk allele calculations can be done. So we'll look at univariate associations. Once we've identified a predictive allele, haplotype context will be explored as a determinant of group status. Then we'll construct a genetic risk score, because you can't look at this entire thing allele by allele. You need to understand how all of the alleles interact with an individual. And these risk scores would account for an individual's risk based on the composite of the implicated neutral and projected HLA alleles and haplotypes. Ultimately, the final testable unit will be an HLA risk score to predict COVID severity rather than the individual alleles and haplotypes, unless perhaps a single allele emerges as a dominant predictor. And the last, and perhaps the most interesting and challenging part, is that the binding characteristics of each of the allotypes in the risk score, positively or negatively associated with susceptibility and with COVID clinical severity, will be determined in an effect to identify the SARS-CoV proteins in their regions that are recognized in aggregate by those making clinically effective or ineffective immune responses. So thank you very much for your attention. And again, thanks to the symposium organizers.
Thank you, Bob. I see there's one attendee. Darcy, go ahead.
So the spread of infections in Africa is just starting. Is there information on allele frequency?
There's a very large effort and a wonderful website that I'll send to you if we get your email, that in exhaustive detail maps the frequency of all of the HLA alleles across the world. It's a huge effort. It's a marvelous compilation of information, and it is available online and you can peruse it. So yes, it's available down to the specific, very specific ethnic subsets of different populations throughout Africa.
My follow-up question is, there is concern about the United States is very diverse ethnically, and there is concern about the spread of the virus and the infectivity of the virus in different subpopulations. And of course, it's complicated by issues of pre-existing conditions. But my thought is there is a database, right? I don't know how good a database it is, where individuals can have their DNA sequence, perhaps not at the level of haplotypes, but they at least would know something about their ancestry. So as this disease spreads across Africa, it will be very interesting to look at the frequency of infectivity and the outcomes across the various African countries, which also are quite genetically diverse depending on the locale.
So Darcy, are you referring to the remarkable increased incidence in African American in the United States?
Well, that's of course what I'm worried about.
Well, that's a very, very good question and observation. And we simply, it is studyable. It's one dimension. I mean, there are many other dimensions that may account for this, socioeconomic ones, et cetera, but certainly the biologic dimension of the difference across individuals comprising the human race is going to be a major factor. And remember that this entire HLA system evolved, and it's perhaps the most complicated, genetically complicated region in the human genome or in the genome of any animal. And it's evolved in response to the challenges of infectious disease. So I think we're really seeing immunology in operation here, and your question exactly gets to the heart of what the immune system does.
Bob, do you know of any studies specifically looking at African American population in the United States, since the prevalence is in some cases 70% in some of the urban areas?
I'm sorry, studies of what in the African Americans?
Well, just clinical studies looking to see if there is a genetic basis for the remarkable increase in prevalence?
No, I don't know of any. Yeah. This is really only emerging now in 2020. So we have some background. There are HLA associations of SARS 1 but to the extent that they can be extrapolated, and I don't think that folks have looked at the HLA alleles that are particularly characteristic of African Americans or different subsets. So it's an open question, I think, right now.
Bob, there's another question from Larry Chasin that came on the Q&A box. He's asking that the SARS spike protein is highly glycosylated. Can the sugar groups be displayed by the HLA route, or is there another mechanism that responds to sugars?
That may influence, usually by the time the peptide gets into the endoplasmic reticulum, it is fairly well deglycosylated. But it's still, there could be areas where, because it's not a human glycosylation, or, well, I guess it would be a human glycosylation. So I imagine all of the human glycosidases would be operative. I don't know specifics, but my presumption would be that they are effectively deglycosylated.
Okay, and there's a question by Raul Rabadan.
So we are very much interested in HLA, and we are looking at this problem. So I sent you an email for potential collaboration. And I don't know if you saw that there is a paper in the bioarchive that they predict for different HLA types. They predict what are the biggest, the better the better binding HLA types to the current virus. And then they were looking at the different populations in the world. And there were several observations about Africans that were quite interesting.
Right. That's really the second paper that has looked at least the potential basis of the diversity of HLA binding of the SARS-CoV-2 peptides. And I think it's going to be an emerging area. I can't emphasize how important the systems biology approach to this question is. And it's something that I would totally welcome any collaboration. We're happy to work with any other people and provide our own immunologic perspective to it. But we really need that type of interaction. It is a big data sort of issue.
There, as you probably are aware, there is kind of big initiatives now to collect genomic information, and HLA is one of the topics that have been discussed. I mean, several consortia international, and also some of the big data sets. I mean, they are starting collecting information about patients in large scales enough to do statistics. So one of the concerns with HLA is, as you know, is that there are many different types, and it's very hard to get the statistics unless numbers are very high. So.
Correct. It is. And of course, locally, I mean, the power of exome sequencing is so great and provides great information. But to try to do the immunologic studies we need to do on viral recognition and the whole issue of what the T cell is seeing, whether it responds effectively by a cytolytic response, or whether it goes into an exhausted mode and sort of ignores virus-laden cells and then triggers a cytokine storm. To understand that, we also need, in rather quick order, HLA typing to work with the very samples that we have in front of us. So it's a kind of, I think there'll be multiple solutions to the challenge of figuring this out.
Thank you, Bob. There's another question by Margaret. Just one second. Margaret, can you?
Yes, can you hear me?
Yes.
Hi, how are you? I'm Margaret Crosby Arnold. I'm a historian. I'm in the history department, so I have been listening in, obviously. And you all, your take of this, of course, is the hard science. But it occurs to me, and I've been hesitant to speak up but it occurs to me as I've been listening, and certainly as this matter has been raised with regards to African Americans or people of African descent, I know that this is not necessarily the focus of you all's focus, but the reality is, you know, you can be walking into a kind of socio-political explosive situation and talking about genetics and all of these sorts of things. My research is focused on the history of the development of the race ideal and all of these kinds of things. And clearly, the priority is on dealing with COVID-19 and figuring out, you know, obviously some way to deal with this medically. But I am wondering how much you all in your work are collaborating with people from the social sciences who might be able to help navigate some of these political trenches or so that you may walk into in dealing with this. I'm thinking in particular about some of the discussions or what Wendy Chung raised with regards to the notion that the deceased are not human subjects, and therefore when you're looking for samples and these kinds of things, if you're dealing with, you know, the deceased who are largely African Americans, because that's who's being affected, how do all of these things begin to play out? And again, here with this HLA and these differences, how are you all thinking about those, or have you collaborated at all?
Oh, it's a extremely important and complex problem that you're addressing. I don't have the answer except to say that from the perspective of someone that looks at HLA genetics, race doesn't have a meaning, but the geographical origin of people really is the meaning. And race is a kind of a construct. It's a very—
Of course it is, yes, of course. And,
yeah. So that,
exactly.
that notion of actually, if you try to publish a paper, particularly in Europe, they would, concepts of race that we would take even as a requirement for submitting to our NIH grants, the racial and ethnic contribution, they would strike that from the paper and just speak about geographical origin. So it's kind of an interesting dynamic that is going on. And yes, indeed, to navigate the cross currents of research in this country now, we certainly need the involvement of people like you that understand the social and historical dimensions of heterogeneity.
That's precisely my point. My expertise is actually Europe. So in looking at this and looking at the construction of race, of course, it's not something that's real in the world. But it is unfortunately ideologically, you know, whipped about in this country in a way that is troubling and problematic. And so what I have been concerned about from my standpoint, and my research standpoint, are the unanticipated collateral damages that come from something like this.
Agree.
Margaret, if I can make a suggestion, because this is obviously moving into a much more complex discussion that can be handled in a Q&A session. So we're going to have future symposia. Perhaps you should sign up to present a little bit of the context and provide some options to people working on this type of problems. Obviously, there's a number of different pathophysiology which have very distinct ethnic heterogeneity. For instance, you know, breast cancer risk in African American women is significantly higher than in Caucasian, and all those have been handled in a very effective way. I don't think they've created, in fact, it's all about providing the best possible care. So I think that if you can provide your perspective, maybe sign up for one of the next seminar slots, could potentially be helpful.
I would certainly be happy to, and I'm happy to collaborate if there are, you know, if there is interest in that.
Yeah. Thank you, Margaret.